in reactions. Catalysts speed up the
Salt,
rate of
pH,
reaction.
and temperature can denature an enzyme.
In general, enzymes are proteins produced by living cells. They act as catalysts in biochemical reactions. A catalyst affects the rate of chemical reactions. One consequence of enzyme activity is that cells can carry out complex chemical activities at relatively low temperatures. In an enzyme catalyzed reaction, the substance to be active on the substrate binds with the active site of the enzyme. One result of this temporary union is a reduction in the energy required to activate the reaction with substrate molecules so that the products of the reactant are formed. This substrate. With the enzyme changing the reaction can be recycled or break down additional substrate molecules. Each enzyme is specific for a particular reaction because its amino acid sequence is unique and causes
The
the active site is the portion of the enzyme is ubiquitous in nearly organisms. One function of the catalase within cells is to prevent the accumulation of toxic levels of hydrogen peroxide, known as a byproduct of metabolic processes. Catalase methods are part of many oxidation reactions that occur in all cells. The primary reaction catalyzed by catalysis is the decomposition of H2O2 to form water and oxygen. 2H2O, 2. The primary reaction catalyzed by catalysis is the decomposition of H2O. The primary reaction catalyzed by catalysis is the decomposition of H2O2 to form water and oxygen. 2H2O2. Yeah, try to cut that out. The absence of catalyst, this reaction occurs spontaneously but very slowly. Catalyst speeds up the reaction considerably. In this experiment, a rate for this experiment, a rate for this reaction will be determined. Once you know about enzymes by studying the kinetics, particularly the changes in the rate of enzyme catalyzed reaction. For example, it is possible to measure the amount of product formed or the amount of substrate used from the moment the reactants are brought together until the reaction is stopped.
sulfuric acid is added to the different cups to denature the enzyme and stop the reaction.
Now we will use titration to collect our data. Potassium permanganate will be added to the 10-second cup. Once all the peroxide has reacted, the
Potassium permanganate will not be decomposed and turn the solution permanently pink or brown. This shows how much substrate is left after the enzyme was denatured.
The enzyme is added to the substrate. Now, and then we're going to do a little transition in there, add a little map. So I'll put E plus O. Okay, after the enzyme is added to the substrate, they bind together. I'm thinking we can, no, start a new one now, so we can be like. After the reaction has occurred, you are left with the enzyme and the product.
For example, when calculating the percent of hydrogen peroxide decomposed after 24 hours,
the new hydrogen peroxide yielded a higher percentage than the. When calculating the percent of hydrogen peroxide decomposed after 24 hours, new hydrogen peroxide yielded a higher percentage than the older hydrogen peroxide. Catalysis or enzymes drastically increase the rate of hydrogen peroxide decomposition. The lab showed how the catalase could be denatured, therefore not increasing the rate of decomposition. The addition of catalase allows hydrogen peroxide to decompose quicker than it would naturally. This is essential to living organisms. The salt concentration can affect the effectiveness.
Amino acid side chains contains groups such as carbolic acid and NH2 that readily gain or lose hydrogen ions. As the pH is lowered, an enzyme will tend to gain hydrogen ions, and eventually enough side chains will be affected so that the enzyme's shape is disrupted. Likewise, as the pH is raised, the enzyme will lose hydrogen ions and eventually lose its active shape. Many enzymes perform optimally in the neutral pH range and are denatured at either an extremely high or low pH. Some enzymes, such as pepsin, which acts in the human stomach where
Generally,
the pH is very
chemical
low, have a
reactions
low
speed up
pH
as the
optimum.
temperature is raised. As the temperature increases, more of the reacting molecules have enough kinetic energy to undergo the reaction. Since enzymes are catalysts for chemical reactions, enzyme reactions also tend to go faster with increasing temperature. However, if the temperature of an enzyme-catalyzed reaction is raised still further, a temperature optimum is reached. Above this value, the kinetic energy of the enzyme and the water molecules is so great that the conformation of the enzyme molecules is disrupted. The positive effect of speeding up the reaction is now more than. However, if the temperature of an enzyme-catalyzed reaction is raised still further, a temperature optimum is reached. Above this value, the kinetic energy of the enzyme and water molecules is so great that the conformation of the enzyme molecule is disrupted. The positive effect of speeding up the reaction is now more than offset by the negative effect of changing the conformation of more and more enzyme molecules. Many proteins are denatured by temperatures around 40 to 50 degrees Celsius, but some are still active at 70 to 80 degrees Celsius, and even a few things withstand boiling. To find the rate of this, you do the change in y over the change in x. So you would do 1.6 minus 1.3 over 60 minus 10. You would get 0.3 over 50. Now put that in your calculator. The answer is 0.006. That is your rate.
Yes.
Many molecules other than the substrate may interact with an enzyme. If such a molecule increases the rate of the reaction, it is an activator, and if it decreases the reaction rate, it is an inhibitor. These molecules can regulate how fast the enzyme acts. Any substance that tends to unfold the enzyme, such as an organic solvent or detergent, will act as an inhibitor. Some inhibitors act by reducing the. Many well-known poisons, such as potassium cyanide, are enzyme inhibitors that interfere with the active site of critical enzymes.